All right, so we're ready. Everybody hearing me?
Yes, sir.
Excellent. All right, so we are, all right, so I'm going to, I'm just fixing some technical issues. Be with you. 1 minute. All right, so it should be all good now. Okay, so the first question is getting a cell, right? That's our first question. And normally it's very regular that you get to differentiate between animal cell and plant cell, right? So here is a real this image was taken from normally a textbook. It more is more reflecting a real cell more than one that you see in your regular CSEC textbook. So this here is your animal cell, this here is your plant cell. So they're asking you what type of microscope would you use to produce images in figure one? So what type of instrument would you get from these images? Anyone with the answer?
Electron microscope.
Electron microscope. Anybody else? Why you say electron microscope? I'm working with the chats too. Okay. Electron microscope. Why electron though? Is there a specific reason why you said that? I just sort it. Yes, you can. Yes, go ahead. Is it? Yes, you can go ahead. Raising your hand, you can go ahead. I'm not hearing because of
Hello?
the Greek. I'm still not hearing. Not sure to
Not
pronounce the name. I'm not hearing. Tyra says because of the great detail. Okay. So yes, we'll indeed use an electron microscope, right? We see that someone said the other types are wrong. No, you need to be a little bit more specific. What are the other types though? You know, you can't just wrong. It has to be a reason. The reason is very important. So put them switch a question and ask you, um, show you a different picture and ask you what are different microscopes. Right? So you need to know the different microscopes, right? And normally use them in your labs, right? You have a compound microscope, right? And we have an electron microscope. We have transmission microscope, right? So the reason why we use an electron microscope, the details and the lighting, right? You can see great lighting details there, right? So ensure that you're familiar with the other types of microscope. But an electron microscope is indeed correct. All right,
Could
moving
you
on to the
repeat
next.
the type of microscopes, please?
You have compound microscope, compound microscope, you have electron microscope, transmission microscope. You get that?
Yes, sir. Thank you.
No problem. Now they're asking you to draw a large, clearly labeled diagram of one plant cell from the microscope, from the micrograph shown in figure 1. So you should draw the plant cell from the figure. So basically, you're going to draw a plant cell, right? Are you familiar with a plant cell? Are you familiar with drawing a plant cell? You should. You should. Diagrams are very, very important when you're preparing for your exams. It's very important that you know how to draw all diagrams, especially cells. All the diagrams in each chapter, you must know how to draw them. So you must practice drawing. Practice your drawing of your diagrams. It's very, very important. Right? So, in the interest of time, we're going to draw it because remember that we have to draw, it says from the labeled figure 1. In the labeling figure 1, so we have to replicate this, right? We have to replicate this. So we can't just go ahead and draw a plant cell that's in our books, right? We'll have to draw it similar to the ones. It's very important that you read the question. So you'll have to draw it similar to this one, right? So you will see where, just going back to the other question, you'll see where the drawing is similar. We have the same 4 chloroplasts here, 6 and 2 chloroplasts here, and we have the same nucleus here, right? So you have to be very, very familiar with your diagrams. We have a YouTube, done a YouTube video on the diagrams, all basically a little crash course on all the diagrams for you to draw, right? So you know you'll have to draw it similar. You have your mitochondria. Somebody can tell me the function of the mitochondria again? I am Who can explain the function of the mitochondria?
It's the powerhouse of the cell.
Yes, the powerhouse of the cell. And what do we mean by powerhouse of the cell? What is that? That's a common term here, powerhouse. What does the powerhouse of the cell have to do with? Why is it the powerhouse? Provides energy. Somebody says provide any site of respiration, right? Excellent. Now, for those at the back, as we know, normally they are kids or students who are afraid to actually tell us that they don't understand, right? Let me break it down. So, as Jimmy said, Cairo, Jamelia, I'm very horrible with names. So if I mispronounce your name, just don't be afraid to correct me, right? It breaks down nutrients to release energy, right? Because here we don't want to look at past paper, we're trying to explain it out, right? So we look at breaking down food. So you know that we break down macronutrients to micronutrients, right? Who can give me an example of micronutrients and its macronutrients, or the macronutrient and its micronutrients? I mean, I talk Spanish. Am I speaking Spanish? I,
No, sir.
I am speaking Spanish?
No, sir.
Oh, okay. Let me speak Spanish, you tell me, you know. All right, so I see Cairo with starch to glucose, and we see carbohydrates to glucose. Right, so carbohydrates and starch to glucose is the same because starch and carbohydrates is basically it can be used interchangeably, and that those are both correct. Right, and then we have also proteins to amino acid. Yes, very good. Right, so all of those are examples of macronutrients to micronutrients. Cool. Now,
Yes, sir.
go again, Douglas.
So I was just saying yes, sir.
Okay, excellent. So that's what the micro, and then now, what are the two type of respiration? Anybody have that?
An anaerobic respiration.
Anaerobic and aerobic respiration. Good. And ensure you know the formulas for both. Right? Ensure you know the formula for aerobic respiration and anaerobic respiration. Do we know the formula?
Yes, sir.
Yes, sir.
Okay, so can somebody type it in the chat? What's the formula, please? Can somebody type in the chat the formula? For those who don't know, because I'm assuming some people don't know the formula. Yes, no? You want me to show you the formula? Are we getting the formula here? The formula, no formula, not seeing the formula yet. I'm seeing somebody said don't remember. Don't remember. Okay. So let me just say that if you have this book, or if you don't have this book, it's one of the best book in my recommendation doing human and social biology or biology. Right? Very, 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 very good tool to use. Right? It's an excellent tool to use. So let me just see if I can scroll quickly. Let me see in the chats though. I'm seeing aerobic respiration, glucose, oxygen plus glucose to form energy, and carbon dioxide. Okay. Do we know the formula of, did I just exit that by mistake? Oh, sorry. Yes, do we know the formula of aerobic respiration?
Yes, sir.
Okay. So here we are. So the respiration, we talk about the form. No, that's not where we are. Here is where we are. So the formula for aerobic respiration is right here. Are you seeing my screen?
Yes, sir.
Sir.
Okay. So the formula for aerobic respiration is glucose plus oxygen. I have to ensure that enzymes in mitochondria. Why enzymes in mitochondria? Because you know what are enzymes, by the way? Let's start there. What are enzymes?
Biological catalysts that speed up the rate of a reaction without itself being consumed or destroyed.
Okay, that's a high flown definition. Excellent. Right? Anybody else? I don't mean catalyst. What do we mean? What's a catalyst?
Anything used to speed up the rate of reaction.
Anything that is used to speed up the rate of reaction. Right? So enzymes are important and enzymes are everywhere, right? And enzymes in the mitochondria, right? Carbon dioxide plus water plus energy, right? And if they ask us to write the formula, you have to know the formula as in C6H12O2, right? Plus 6O2 over enzymes, mitochondria, and etc., etc., etc. Cool?
Yes, sir.
Nice. So the name of the book is The Concise. Nice.
Sir,
Excellent.
what page is that?
This is page 64. Right? And there's also the anaerobic formula, right? And you also need to know that as well, just in case. You have two of them. Well, it's two of them. Yes. One is anaerobic respiration in yeast, and the other one is anaerobic respiration in muscle cells. All right? The ones in the we are familiar with ethanol, right?
Yes, sir.
Right. So ethanol is a type of alcohol, right? That is the end result of yeast. That's why they use yeast in fermentation, right? So yeast fermentation process produces alcohol, carbon dioxide, and two ATP molecules, right? And ATP stands for, if you don't know, adenosine triphosphate, right? And then lactic acid. We know that is what explain how lactic acid is formed. Somebody. How is lactic acid formed?
During anaerobic respiration, the glucose doesn't fully break down, and well, due to it not being completely broken down, it creates lactic acid, which in turn causes muscles to become fatigued and slow down, which would lead to an oxygen. Or I'm going too far.
No, no, it's fine. It's fine. But for the people at the back, a more simpler one would be how is it formed, right? So basically, we know that we should warm up before we do any major exercise. Cool. So warming up before you do any major exercise, that is to allow oxygen to flow in our body. The more oxygen we have is the more chance for aerobic respiration, right? Anaerobic respiration is really when the body don't have enough oxygen to break down the food necessary to give us energy, right? Because remember, aerobic respiration is using oxygen to break down molecules to release glucose, which give us energy. Am I going too fast for the people at the back?
Yes, sir.
Okay. So if I'm going too fast, let me know. And it's very important if you don't understand to let me know. Okay. So let's say that you are going to run a race, right? You ever start run a race or you ever move up some step and you start feeling breathing heavily? Anybody ever
Yes.
experienced that before?
Yes, sir.
Why you start to breathe heavy is because your body needs oxygen to give you that energy to go up the stairs. Are you following me?
Yes,
Yes,
sir.
sir.
So your body need oxygen to break down the food in order for you to go up the stairs. So the body will break down, the body use oxygen to break down the food molecules, which is more than likely carbohydrates, to get glucose, which is sugar, and that sugar results in energy. Am I with you so far?
Yes, sir.
The people at the back, I'm speaking to you now. Am I with you so far?
Yes, sir.
Okay. So what happens is that the body when you are going up the stairs, the body don't have enough energy, don't have enough oxygen, right, to break down that food molecules. Oh, cool. So therefore, what happens? What happens is that the body now start to use anaerobic respiration, right? Because it doesn't have enough oxygen. And then as a result of that, we see where it starts to create its own energy, right? So it starts to just break down the glucose. Because we can see here where where aerobic respiration uses, and let me just get the annotation here. We see aerobic respiration uses glucose and oxygen. But for anaerobic respiration, just use glucose, no oxygen, right? So when you're going up the stairs, there's there's a lack of oxygen. So the body say, yo, you hear me I say, we don't have the oxygen, so let's create some energy on our own. So it uses the glucose and the enzymes in the cytoplasm. And for all who did not know, the cytoplasm is the main source of where all chemical reaction take place in the cell. Let me say that again for the people at the back. The cytoplasm is where all, if not most, chemical reaction take place in the cell. All right? Even though this is for yeast. But the one we need to talk about would be the one in muscle cell. So we see where glucose alone is used, enzymes in the cytoplasm produces lactic acid and energy. So what happens is that it gives you energy, yes, but lactic acid becomes a byproduct. And lactic acid causes discomfort. It causes pain in your muscle. Right? So that is why the safest way or the better way, let's not use safe, the better way would be using energy, which is aerobic respiration and not anaerobic respiration, because it gives you a byproduct of lactic acid, which is not usually good for the body. Are we good? Is there anyone that's lost?
Yes, sir.
So we lost or we're good?
Good, sir.
Good.
Okay. So Sue, no, this is not the only marathon. We're having other marathons coming up, but we have also extra classes which are paid otherwise. But I'm just doing something for the people who probably can't, even though the paid is cheap. The paid is just $1,000 per class. All right, but I'm still doing something for free, which we do every year. All right. So let's jump back to the question. So we understand aerobic respiration so far. Good. So let's jump back to the question. You see, sometimes one
Excuse me,
question can.
sir.
Yes, go ahead.
Sir, what year past paper are you doing now?
This is 2023.
Okay, sir.
Okay. So we talk about the mitochondria and what is the we see there is a nucleus here. What is a nucleus though? What is this? What is this here? Anybody know it? What's that?
Sir,
What's
does
this?
that contain the DNA for the cell?
So we said this contains the DNA of the cell. Okay. Anybody else? If we are told to draw the nucleus, could you draw it?
No,
Like a up close version of the nucleus. Could you draw that?
Probably not, sir.
no, sir.
Because it's very important that you can, always, always you can't be too prepared. All right. So let's jump back to my favorite book. And we go up to cells. So we can zoom in. Let me see if I have a zoom function here. Yes, nice. And let's talk about the nucleus now. It's very important that you can draw the nucleus, right? We can see there's a barrier here where we have the nucleus membrane, we have the nucleoplasm, right? And we know that plasm is really water or any jelly-like substance, right? So that's why you have the cytoplasm. So biology is pretty simple. It's mostly a prefix suffix kind of thing. So if you know what the prefix mean, you know what the suffix mean, then you can put it together. Right? So what cyto, anybody know what cyto mean? Cyto means cell, right? And plasm is water. So basically it's a jelly-like substance found in the cell. Right? Then now we say nucleoplasm, and plasm again is jelly-like substance. Nucleo has to do with the new data, you get jelly-like substance found in the nucleus. Right? And then we have the chromatid thread. And the chromatid thread is basically what DNA is all about. Right? Chromosomes, chromatids. And then we have the nucleus. Right? And then all of these four parts combined give you the nucleus. Did we have this picture here?
Yes, sir.
All right, so
Yes,
ensure
sir.
that we can draw this diagram. Cool? Then we look at the plant cell and it's pretty much the same except for the chloroplast and the rest of them. What's the chloroplast? Function of a chloroplast?
To absorb the light
So-
energy from the sun.
To?
Absorb the light energy from the sun so that the plant can make food.
Okay. Anybody else? Come on, let's put some life in it, man. It's free, energy, education can't bore, Let's go. What else?
This is where photosynthesis takes place.
This is where photosynthesis takes place. Okay.
It
There is a green pigment.
contains
Green pigment. Is it the green pigment though?
it contains the green pigment.
Okay. Now you talk. Now you're talking. Right? So this is the site where photosynthesis occur. Right? And use sunlight to make what?
Energy.
Our food. Yes. Cool. So can somebody tell me now? Let me see which people bright now. I see what thing. Look at the anaerobic formula. It's the opposite of which formula?
Photosynthesis.
Photosynthesis formula.
Photosynthesis. Excellent. So if you know photosynthesis formula, you know anaerobic, you know aerobic, sorry, respiration formula, We never see that.
Yes, sir.
The people around our back, the back people, them know. You never see that.
Yes, sir.
All right, make sure. But just because we know some people now go talk, we're going to look and see. So what's the formula for photosynthesis? Anybody know? Let me see if I can find it fast.
6 CO2 plus 6 H12
Okay.
forms C6H12O6 plus C plus 6 O2.
All right.
And the
Nice.
process of sunlight and chlorophyll, sorry.
Okay, nice. All right. So let's see if I can get a visual representation of the formula here. Here is the formula. So the formula is carbon dioxide plus water, energy absorbed by chlorophyll, glucose plus oxygen would be the product. Right? And then we looked at aerobic respiration. We see where glucose and oxygen, we see where it flip over. So we see where glucose is now replaced here and oxygen is now here. And then carbon dioxide becomes the byproduct. Right? So if you know that one, you know the other. Excellent. All right, so let's move into what's the function of the vacuole?
To store minerals and water.
To store minerals and didn't hear the last part.
Water.
It's not exactly water though, you know.
Sir, it stores food. That's where the food is stored.
You said store food.
It stores the products of photosynthesis and dissolved minerals.
Dissolved minerals, yes.
It contains
Yes.
cell spot.
Hmm. All right, so let's look at it, guys. It's not necessarily water, you know, but I want to be as precise as possible. Right? So it's not necessarily water. Sorry, let me see what's going on here. Stop sharing. Oh, my bad, guys. Okay, so let's run back up to cells. So you have to ensure that these, you know them. Cool? So let's look at the vacuole. The vacuole has a large membrane-bound compartment containing cell sap, as somebody correctly said. So it's not necessarily water. It stores water, you know, but it's not water. It's not water alone. It's a mix of sugars and mineral salt and amino acids and waste products. Right? It says ensure when you talk about the function, you say, boy, you want your function to be as specific as possible. So a large membrane-bound compartment containing cell sap, a solution of sugar, mineral salts, amino acid. Cool?
Yes, sir.
All right.
Yes, sir.
Yeah, man, I love one, you know, I love distinction, you know, so me always I go push it to be specific, you know. So yeah, that's just me, guys. All right. Think we're good enough here. Let's move on. So the next question states, somebody can read next question for me.
Yes, sir.
Two similarities between the plant cell and the animal cell.
I will know that soft. Who want to take that?
They both
On your
have a
three.
cell membrane.
They both have a go ahead. Nichelle? Is it Nichelle?
Chanel, they both have a cell membrane.
Oh, they
And
both have a cell membrane. Okay.
Animal cells do
Yes.
not have a cell wall and plant cells have a cell wall.
Yes. Which bright somebody can tell me why animal cell don't have a cell wall.
Because if the whole point of a cell wall is no, the cell wall is fully permeable. So if if the if it has a cell wall, it's going to become rigid.
That question is tough, though. I never think about that one, they done. Anybody else who I appreciate the effort. Let me checking at the chat. Sir, because it needs diffusion to take place. Nah, you're not wrong, it's not the main reason. One of the main reason. Yes. Anybody with the main reason.
Because the cell wall is made out of cellulose and animals don't have that.
But that's a brilliant fact, Which school are you? Are you from Jamaica, Steer?
Yes, sir.
Which school are you from?
Mobe High.
Mobe. Okay, big up Mobe. That's a brilliant answer, but it's not the answer I'm looking for. But it's a brilliant fact. The cell wall, how much of you guys knew that the cell wall was actually made up of cellulose? Right? It's an
Yes, sir. I know.
yes, so who never know? For the people on the back who never know, write that down in your brain or jot it down in your notepad. It's made up of cellulose, and cellulose is a type of sugar, but it is a macronutrient. It's not broken down. It is the highest form of sugar that's not broken down. That's why it's not sweet. Excellent,
Excuse me,
Kyle.
sir.
Yes, sir.
I think the animal cells don't have cellulose because it's made up of a compound that is very rigid, so it would not allow for proper movement in animals.
That is a great point. Which school are you from? Are you from Jamaica?
Yes, sir. I go to Monroe College.
Monroe College. That's big up Monroe on a St. Best. Excellent, excellent answer, but not the answer I'm looking for. The actual question was, why does a plant, what's the main reason why a plant cell does not, why an animal cell doesn't have a cell wall? And you'll be surprised to know, that the main reason why is because animal cells don't have a skeleton, and without that skeleton support, they cannot stand upright without the cell wall. That's a fun fact, right?
I don't understand.
You don't understand that? Okay. So what makes animals stand upright is because of the skeleton structure. The skeleton structure that your bones make you an animal, humans, dogs, cat, they can stand upright. But for plants don't have a bones. So how would you see plants that are upright, even though they have the root alone can't allow you to stay upright. They would just be in the ground and everybody that just drop. So plants would be like this. Let me see if I can draw it. The plants would be like this going up and then it just fall back down. You understand? So it's a fun fact to know that the cell wall keeps the plant upright because they act as the skeleton. They do the same job as the skeleton system does to the animal. That's what the cells of a plant do for them. That's a fun fact. The first to the last, everybody get that? I'm
Yes.
still talking Japanese.
Yes, sir.
Oh, so English now. It's not Japanese.
No,
Excellent.
sir.
All right. So yes, two similarities between the plant cell and animal cells. Membrane-bound nucleus, which contains DNA, right? Plasma membrane or cell membrane. Cytoplasm, in which other organelles are suspended, right? And we also have an endoplasmic reticulum. What's the endoplasmic reticulum? They both have endoplasmic reticulum. What's that?
It's a structure that helps to transport the proteins that the ribosomes would make.
Oh, that's a beautiful answer. Yes, Mobi. Mobi in town. I like that. Yes, beautiful. So, for those who are at the back, right, ribosomes make protein. Did we know that? Or synthesize protein. Did we know that?
Yes, sir.
So when they make that protein, endoplasmic reticulum will transfer it where in the body where it is needed. Yes, Harriet.
Yes, sir. Aren't there two types of endoplasmic reticulum, like the rough one and the smooth one? I think one transports protein and the other transports lipids or phospholipids.
Yes, sir.
But
Because the smooth one would be without the ribosomes. That's why they are called smooth endoplasmic reticulum. Where endoplasmic reticulums are normally dots, and they appear to be dots in an electron microscope on the endoplasmic. Right? So you have smooth reticulum and you have rough. The rough one would be the ER, the one that has ribosomes. Cool? I will ask, everybody get that? Is there anybody who don't get that?
Sir, can you repeat?
Okay, so I'm saying that there are two types of ribosome. They are rough and they are, when I say ribosome, sorry. There are two types of endoplasmic reticulum, rough and smooth. Let me see if I can, let me see if there's one in the text. I'm not sure if they gave this one in the text, did they? Did they? No, they did not. So there's rough and smooth, right? The rough one, let's jump back to the question. Right. So let me try and annotate it. The rough one, there are two types of endoplasmic reticulum, smooth and rough. The smooth one would be like here, just smooth. The rough one would have dots on it. A lot of these dots. The following. So
Yes, sir.
these dots on it would be called the ribosomes. And these ribosomes are what makes protein. So there are two types. And we call this smooth E, SE, that's smooth endoplasmic reticulum, and this is rough endoplasmic reticulum. So smooth ER and rough ER. And rough would be the ones that has ribosome, but it has all those dots on it. Make sense?
Yes, sir.
Excellent. Then the next one says the most prominent structure on the animal cell electron micrograph carries genetic information in the form of DNA. State the name of this structure.
The nucleus.
Nucleus.
So we have the nucleus as the answer there. Anybody else is something different than the nucleus? Do we have something different? No? Yes? No? Yes?
Third.
I don't think we have something different.
No, it's not. It's the nucleus. Right? Excellent. So the nucleus is responsible for genetic information. Moving on. Diagram in figure 2 shows general structure of a bacterial cell. And also you need to know how to draw an algae. You need to know how to draw an algae and draw anything you need to draw, you need to can identify. So draw an algae, draw a bacterial cell, all of these diagrams you need to know how to draw them. Right? And you need to know the difference between bacterial cell and all animal and plant cell. So they state that this is a bacterial cell and to identify one similarity between bacterial cell and plant cell.
They
What
both
do
have
you?
cell wall.
They both have a cell wall.
They
Okay.
both have a cell membrane.
They both have a cell membrane. Excellent. Anything else that they both have?
Cytoplasm.
We see that they have a cytoplasm.
They also have genetic material.
Yes. And they also have ribosomes. All right. So let's see if we can check my favorite book and see what's there. And we see that we need to know the bacteria cell. Right? And this is the bacteria cell. So the bacteria cell has an extra layer on it called the capsule. And the capsule is normally the slimy part. It's very slimy. So it sticks on the surfaces. That's why you can never get rid of bacteria 100%. You ever hear that term that you can never kill bacteria?
Yes, sir.
You notice products always say 99.9% effective, but never 100%. It's because of the capsule. The capsule is the outermost layer. So it has three layers. It has the capsule, then the cell wall, then the cell membrane. And the capsule sticks on the surfaces. It sticks on the surfaces when you're trying to clean. It's hard to remove. Then we have the cell wall behind the capsule, right? Then we have a cell membrane, right? It doesn't have a nucleus. It has a nucleoid, right? So yes, it has. The person who says nucleus, you want to go with something safer because it doesn't really has nucleus. It has DNA in a sense, but the others would be safer. You want to go to this in a last resort, right? It has a cytoplasm, right? And it has something called a plasmid, which is another piece of DNA, right? So it has two real pieces of DNA, the coiled one, the long coiled one in the middle, which is the nucleoid, and the plasmid, which is a circular piece of DNA here. And the flagellum allows it to move. So are we familiar with this? If we're not, we can take a picture, a screenshot of it, draw it. If we have the book, we make note. All right? Comfortable?
Yes, sir.
Okay. So yes, any one of these cell wall, cell membrane, ribosome, cytoplasm, all of them would be accepted. Moving on. Next question says the structure of the bacterial cell are especially suited for its role in infecting other organisms. Complete table 1 by explaining how each structure helps the bacterium to affect organism. So remember now that you now are talking about how does, not to explain the functions, read the question properly, is not to explain the functions just so, it's to explain the function as it relates to affecting other organism or infecting other organism. Yes, Harriet.
Yes, sir. The flagellum would help bacteria to infect an organism by increasing the speed at which it moves, because the flagellum helps the bacterial cell to move like, I don't know how to explain it properly, but it helps to move more efficiently.
Okay.
Yeah.
All
So
right, all right.
it's
So
like swim.
Or for
that's a
something,
just before you continue, let's hold that thought. Um, that is it. All right, that's another thing I wanted to talk about too. Biology, don't overcomplicate it, all right? Some of the answers as easy as you think. You see me? So yeah, try and figure it out, and you can get it simple. You see, last part you say help to move efficiently, you just miss something. Just something. Let me see who could have fix up that.
sir, so a better or a shorter statement, you could say that enables or it enables movement towards the target or the host cell. You say that?
Yes. Yes. So that would have give a nice 2 marks. All right. So you just say helps movement towards the host or the target. And you're done. That's an efficient mark. All right. So next one, the capsule. I can't explain that already still, but let's see how it goes. Yes.
So it protects the bacterium from harmful cells, and it also helps the bacterium to stick to the target cells.
To stick to the target cell. Yes, we'll take that. So just to explain, Cherika,
Yes, sir.
which school are you from, ma'am?
I'm from the Pembroke High School.
Big up Pembroke. Yeah, represent well. Is there any of your classmates on here?
I'm not sure. It's supposed to be a group of us, but I don't really see anybody else.
Most of them bail player. Gotcha.
And to say this, I was really wondering if there was a class out there and somebody shared the link. So I'm expecting the person to be here with me, but I guess I'm by myself.
I say go, man. Me tell you how it a go on now. You see when is the exam again? Is it I don't remember the date. Somebody have the date?
I think it's the first exam is in May. I'm not
No,
sure.
the bio paper 2. When is that?
Yeah, May 19th.
So
May 19?
Yes, sir.
All right. Yeah, and I really May 18, May 18 the place I got stuck full. I'm telling you, we do this every year. Like, you see, the week before, it's going to be everybody that's gonna try to cram it in. So it's good that the 30 of you guys that are here, or 33 guys that are here. Normally, the ones that come out early are the ones that get the one. Morris,
Yes, sir.
when I hear your fan day, my boss, you good?
Yes, sir. Me just a listen and observe.
Oh, so you know what, chime in for some of the answer them, man.
Sir, me not really good from bio like that. We just a try refresh.
No, man, but here what go on now. You see, if you say foolishness, Morris, I know you have to say it, Because once you say it now, you have to remember when you say it and when I correct you, you're better off.
Yes, sir. That's good. That's
So
good.
that, which school you from, Morris?
Calabar, sir.
Calabar, big up bar. Ratabar. You still call it Ratabar? I'm old.
Sir, that might be a little bit too old for me,
Oh, I'm old, Jaja.
Yes,
Alright.
sir.
Alright, good. So the capsule, remember we say it's a remember I said it's an extra component. So it's slimy, it helps to stick. And what it would also do, I could go back to it because we know some people not like hearing me talking and them like see the visual. So if we go back to it, we see that it's an extra component. So what now? What to when we use a Lysol and start spray? When we use a Lysol and start spray, it protects the bacteria too. Because the Lysol have got pass through the capsule, then it have got pass through the cell wall, and then it have got pass through the cell membrane. So it has to pass through 3 layers. So that means the capsule will protect any chemical substances from attacking the bacteria itself. How my sound? How
Sounds good,
sound?
sir. So
Yes. Go ahead.
the capsule will be the thickest layer, right?
Yes, yes. You can see it there. It's thick. Yes.
Excuse me, sir.
Go ahead,
Is it
Miss Lee.
is it semi-permeable or permeable, like
Which one?
the capsule?
No, it's not permeable at all.
Okay.
Yeah, but you know that chemicals can penetrate.
So
But
nothing enters the cell?
all right, so when we talk about permeable, we're saying that on a normal circumstances, it's not. But if a chemical, you know that chemical would eat its way through.
Yes, sir, it would.
Right. So permeable and semi-permeable would really mean that under normal circumstances, can substances go through it? Right? But a chemical is really not normal circumstances. Okay?
Okay,
Miss
sir, thank you.
Lee, what are you here all along?
No, I wasn't. I just joined. That's why I'm probably not understanding. I'm sorry.
No, you ask questions just because I didn't hear you participating before. Welcome. Which school are you from?
Caribbean Online Academy.
They go ahead. Which one?
Caribbean Online Academy.
Oh, first I'm hearing that one. Where is that located?
It's an online school. That's why it's Online
Oh,
Academy. Yes.
oh, it's an online academy. Which country is that? In Jamaica or somewhere else?
It's regional. So it's the
Oh, it's
Caribbean.
I'm hearing that. Thank you for that information. Are you from Jamaica?
Yes, sir, I am.
Okay. All right. Big up the online Caribbean school. All right. So yes,
Thank you, sir.
so the capsule is a slimy, so we get that. All right. Let's move back to the question. So we will say that the flagellum aids in the movement towards the host, and then we say the capsule protects the cell from chemicals, allowing the cell to stick to the host cell. And what's the purpose of the cell wall? It's kind of similar.
To stop the bacteria from bursting when water enters the cell, or acts like a tough outer layer that protects the bacteria from danger.
Under normal circumstances, yes, but in this context, you want to get full marks.
It
So the full
protect.
context would be
It
that.
provides.
Mm-hmm.
It provides protection from antibodies.
Okay. 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 I like that one. Who said that again? Erika?
Yes, sir.
Frembokal? Frembokal, tough man. Yeah, I like that. And let me see if you're really tough. You ready for your test?
Sir, we're not talking about 1 minute over.
Let me see if you're tough with you. Which antibody would it protect it from?
Sir, come back to me. Come back to me.
Sherry, come on. Frembokal, come on, man. Which antibody would the cell wall protect the the bacteria from? Joe, this is the white blood cell. You're close. Which one of them, Jody? Which one of them? Phagocytes. This is the answer. Yes. So the phagocytes. And what's the role of the phagocytes? May I push on you know? May I push on sign? What's the role of the phagocyte? Who can tell me that?
They
Caribbean. Yes.
they release chemicals that break down the bacteria so that they can't harm you.
Who is that?
Cairo.
Cairo. Not necessarily.
Sir, are these the white blood cells that engulf and absorb the,
And what engulf mean?
so they consume or they absorb the bacteria?
Yes. Yes. So the phagocytes, so there are two types. Phagocytes, who know the other one?
Lymphocytes.
Lymphocytes. So the phagocytes are the one them will go out there and fight. The lymphocytes, sorry, are the one them will go out there and war the bacteria. We always talk about that. Right? Them when they go out there and then war them physically in layman terms. You know, when we are going to school, that's how we get students to understand. So we go so boom and them go out there and war. While the phagocytes eat. So them do the cleanup. Right? Yes, Douglas.
Sir, what my teacher would always tell me that it's basically like the JDF and the JCF.
Yes. So the JCF, yeah, that yes, it does work. But unless I say JDF people. I heard that one too, but me not too like it because the it's valid enough. It's just my personal opinion. I mean, I think the JDF eat people. Yeah. So the engulf part, which is the most important part, right? So eat people, yeah. So unless I go put it and say the JDF eat people, then we can work with it.
Say the
You know what
JF eat people, is that what you said?
Yeah, I mean, let's make up some things so we can remember it, you know? So I mean,
Okay, sir.
if we're gonna work with JCF and the JDF, and we are say lymphocytes are the JCF, and we're gonna say that the phagocytes are the JDF, then we're gonna say JDF then basically eat up the people, engulf the people. Right? So
Sir, just from memory.
huh?
Just use it from memory.
Yeah, man, it's just from memory. It's just, that not make what we are saying don't make no sense. It's just from memory. Sometimes I just make up some things so we can remember. Alright? You can't lame in the moment studying becomes lame in the moment. Once you take out the book, you just step on, you just read. It will become lame. Like, me not like teach lame. If me there sound and not talk to me, I go end the life. That's me that. I just not like lame. I like conversations and talking and them thing that make it remember stuff. Alright, let's look at the next question. Next question states humans. Human beings are multicellular and are made up of large variety of cells. Complete table 2 by distinguishing between the following cell: red blood cell, skin cell, and nerve cell. All right. What? Yes, Pembroke Hall. I see you open your mic.
Um, so the red blood cells and the skin cells. Red blood cells have no nucleus, while the skin cell have nucleus.
So the red blood cell has no nucleus.
And
So the red blood cell has no nucleus and the skin cell has nucleus. That's what you say?
Yes,
Big
sir.
answer, but you're mega pusher now. You ready for it now? You ready for it?
No, not this time. Get me next
Yeah, man,
time,
ready
please.
for it, man. What you know now? Why does the red blood cell doesn't have a nucleus?
To allow easy so I can't remember that.
And sir, could I answer that
To
question?
maximize the amount of
All right, Miss Caribbean Academy.
Sir, a red blood cell doesn't have a nucleus for two purposes. One, so that it's interchangeable, because when it doesn't have a nucleus, it shortens its lifespan. And two, so that it can allow as much oxygen and nutrients and I guess hemoglobin that can go into the red blood cell. I'm not sure.
All right, Caribbean, like you're thinking. It's basically for that purpose. The main reason for a red blood cell not having a nucleus is the space. Because the main purpose of the red blood cell is to transport nutrients and, most importantly, oxygen around the body. So the space for the nucleus are got there. It are got limit the amount of oxygen it can carry.
Sir,
That make sense?
also, yes, sir. Also, another difference is that red blood cells have hemoglobin. Do skin cells have hemoglobin? I don't think so.
No, man, because the skin cell wouldn't basically carry no blood. Excellent. That's also a valid answer. Right? So the red blood cell, the first answer by Miss Pembroke was spot on. Second answer by Miss Lee was spot on. And just to note that they can ask you this. So normally we not try to swat past paper like that. We try to reason it out. So if it come down a different way, we can act it same way. So red blood cell doesn't have a nucleus because it limits the space where it's supposed to take, where it's supposed to carry oxygen around the body. We consider the red blood cell like a bus, and we consider oxygen like people, right? So the seat, the seat actually limit the amount of space
Sir, I
that
have
the
one
bus
more
can
structure.
carry. Huh?
I have one more structure, sir.
Yes, go
Um,
ahead.
the red blood cell also has a concave shape, to increase surface area, and the
Indeed.
skin cells wouldn't.
Caribbean people have it, man. Yes. So all of these answers are good. So biconcave, this shape consists of hemoglobin and they have no nucleus, while skin cell has a nucleus and they are not biconcave. And they are biconcave because they increase the surface area. What does surface area mean? It increases the space that the cell has in order to trap oxygen to carry it around the body. Anybody lost or we're all good?
Some
sir.
laws.
All right, so yes, me still pay attention to the chat, guys. Yes, me see the chat. Jimmy, Williams, you're all correct. Nice. Then now, what's the difference between the skin cell and the nerve cell? What we see who have that?
The skin cell is flat. I think the term that we use is plate-like, and then the nerve cell is long and branched.
All right. Miss Grant,
Yes, sir.
which school are you from?
Montego Bay High School.
Montego Bay High School. Big up Moby. All right. Yes. Somebody can give me another reason? Whole heap of them, guys.
Sir, the skin cell would have dip on the outer layer and the nerve cell would have dip under the skin.
All right, location. But we're not talking about location, Morris. They say that lose mark. No, we're talking about structure. So it wouldn't be location. It would have be structure, and structure mean how they are, the things that they consist of. Right? How different they are in terms of structure. So structure means if you put two person together, you're gonna say, all right, then one fat, one slim, one head big, one head little, one nose big, one nose little. Cool? It's a structure.
Sir, can I try?
Go ahead.
So the skin cell is flat, while the nerve cell is long and thin.
Okay. But we are going hard though. It's easy, guys. Think about the nerve cell. What does a nerve cell have?
It has like the axons and the axon terminals and all of those things.
So why not use all of that? Nerve cell has axons and myelin sheets and dendrites and all the what the other name? What the other one name? Oh man. Where that go, Where that go? Who can tell me the R word? What name again? Ah.
Receptors.
Node of Ranvier.
Sir, the
That.
receptor?
Nodes of Ranvier. Have we ever know nodes of Ranvier?
Yes, sir.
No, sir.
Yes, sir.
So, nerve cell. Are we comfortable with the nerves?
Yes and no, sir.
Yeah, that's another thing that people hate, the nerve. I don't know why you guys hate the nerve so much, man. Nerve cell consists of myelin sheet, nodes of Ranvier, axia, and dendrites, while the skin does not have these structure. All of that simple. That's all. Yeah, man. Simple 4 marks, that guys. You want us to touch on the nerve?
Yes, sir.
Because I know the nerve normally the last place in the syllabus and oftentimes it's not completed. So let's check the nerve cell. It's cheesy, guys. Cheesy, 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 cheesy. Let's see if we can get the yes. So guys, if you want to know anything, just hit the diagrams and you're good. You can study biology just from the diagrams. Oh man, like if you know the diagrams and know what they are, the functions, you're good. All right, so we have the myelin sheet, and the myelin sheet here is to protect the cell. You see, it look like what does this drawing reminds you of? The myelin sheet.
Almost like the capsule in bacteria cells.
Yes, the capsule in the bacteria, right? So the myelin sheet insulate, right, and protects the nerve fiber and speed up transmission of impulses. Do we know what are impulses?
Yes,
Sir,
sir.
is that the reactions like
So impulses, we say reactions, what else? Right, so we say electric shock. So impulses are basically the sensation, the feeling. So the touch, right? Anything that you're feeling. So let's say you step on a needle, that's the impulse, right? Let's say you touch
It's
your hand going on the
basically
fire.
transmits messages.
Go ahead.
It transmits messages.
Right, so the impulses are the messages. Excellent. Thank you for that term. Nice, the messages. Right, so whether cold, hot, whatever. And these impulses travel along the dendrite. Right? So let's start off first. The receptor, what is the receptor?
The area on the nerve cell that detects stimuli or will detect the impulse.
Yes, so the area and the message that detects the stimuli, and the stimuli will be the whatever feeling that is. Right? And give me
Changing
an
the environment, whether internal or external.
excellent. And give me an example of a receptor.
The skin.
The skin. Excellent. So we see where the skin now is attached, and we see now where the receptor is attached to the dendrites. Right? And the dendrite is the long, see this here, this long structure here? What's a dendrite? Who can tell me that? If I should define the dendrite, how would you say?
The area on the cell where the impulses travel.
The area on the cell where the impulses travel. Nice. Nice.
Sir, the denture, it's like nerve fiber that carry impulses towards the cell body.
Nice. It carry the impulses towards the cell body, and we can see that's what it's doing here. Right? And then now we see that here is the node of Ranvier. And the node of Ranvier would be any constriction. And constriction, any as when the gaps in between. Those are what the node of Ranvier are. And then go ahead, Douglas.
Yes, sir. So if there's a case where they ask us for the function of the dendrites or what are the dendrites, do we have to put electrical impulses or we could just put impulses?
Yeah, I mean, yeah, I don't think it's hard and fast.
Okay, sir.
Right. I don't think it's hard and fast. But if they ask you now, what's the difference between the axon and the dendrite, what did you say?
Impulses travel along the dendrite cell body, and then at the axon, the impulses move to another nerve cell.
Okay.
Are the nerve cells located in the skin as well, right?
Nerve cells are in the brain and the spinal cord.
So what transmits stimuli to the body through the skin?
Receptor. The skin is a receptor.
The axon is basically the same thing as the dent. I can't pronounce it. It's the same thing,
Dendrite.
but yeah, it's the same thing, but the dendrites carry towards the cell body, and the axon carry impulses from the cell body. So
That
it's
is
just
exactly correct. So the dendrites are receivers. The dendrites receive the message. The dendrites receive the impulses. You can see it here. It's taking it from the receptor and it carries it to the cell body. Right? And then the axon now takes it from the cell body and takes it to the synaptic knobs. Right? That would be another cell. Nerve cell, that is. You understand that?
yes, sir.
So one takes it from and one takes it away. So one takes it to the cell body and one takes it away from the cell body. Is there anyone that's lost?
Repeat, sir.
So the difference between the dendrite and the axon. The dendrites take impulses from the receptor, and the receptor can be a skin, right? And takes it to the cell body. Let me annotate so you can understand. So annotate. Here we go. So the dendrite takes it from the receptor. You're following me?
Yes, sir.
To the cell body. So it receives information from the receptor to the cell body. Right? And then now the axon takes up that information from the cell body. Let me use a different color. Takes it from the cell body to another nerve cell. Right here would be another nerve cell. basically. So it takes it away from the cell body. So one receives the information, receives the impulses, takes it to the cell body, while one takes it away from the cell body. So the axon takes it away from the cell body, while it then tries to take it to the cell body. Are we good?
Yes, sir.
Yes, sir.
Yes, sir.
Is there anybody that's not good? I take silence as a good. Now let's move on. Let's look at I forgot I have to take off this. So just to point out something at the top here. We have two neurons are made up of both CNS and PNS. Right? CNS is what does CNS mean? Central nervous system. Right? And the central, and then PNS with the peripheral nervous system. Cool?
Yes, sir.
And they transmit messages called nerve impulses. All neurons have a cell body within with thin fibers of cytoplasm extending blah 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 blah. Right? And then axon blah 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 blah. So CNS, PNS. Then you have the sensory neurons and the motor neurons. Sensory neurons transmit impulses from receptor to CNS. So from the receptor to the central nervous system. And it's very important that you know that the central nervous system is made up of the brain and the spinal cord. Right? And then motor neurons transmit impulses from the CNS to the effectors. What are the effectors?
Sir, that's the part of the organism that responds to the stimulus.
Right, so it responds to the stimulus. So what happens? Let's look at it now. So the receptor is the steps of the skin, right? And let's say there are some fire here. Let's call this fire, right? And then heat. So the heat now is transmitted along the sensory neuron, right? And then now it goes to the CNS. What does CNS mean again?
Central nervous system.
Central nervous system. And the CNS is made up of what again?
The brain and the spinal cord.
All right. And then now we have what is a relay neuron in the middle. Now, relay neuron doesn't have a myelin sheath. Now, let me see if there was something that was before here. Let me see. You could go here. Sorry, let's go back up. Let's go back up. No. Okay, so why does the relay neuron doesn't have a why does the relay neuron doesn't have a myelin sheath? Why you think so? Why you think the relay neuron doesn't have a myelin sheath?
Because the purpose of the relay neuron is just to transfer the impulse or the message from the sensory to the motor. So the impulse is not staying within the relay that long that it needs to be insulated.
And it travels faster without the myelin sheath. Well, does it? Yes, it does. Cool?
No, sir, explain why.
Does it? I'm asking you.
No, because you said that the myelin sheath has to speed up the transmissions, which would be the impulse. So then
Right.
if it doesn't have one, then why? I mean, it makes It's that it has to be fast in order to transmit from the sensory to the motor. But but why are you saying that without the myelin sheet it would
No,
be
I'm
fast?
asking you.
But I don't understand.
Yeah, I don't understand. That's why I'm asking you. I'm asking that question. Does it travel faster without the myelin sheet? Yes or no?
I would say no.
You say no. Okay. Anybody else? That's an interesting question. Does it travel faster without the myelin sheet? Because the myelin sheet, one of the purpose of the myelin sheet is to speed up the transmission of the impulse. I've never thought about it, What says you as biologist?
Sorry, it would not go faster without the myelin sheet. It would slow down.
Okay. It does travel faster without the myelin sheet, though. In the case of a relay neuron, it travels faster without it. But the sensory neuron does need that protection, while the relay neuron doesn't. Why you think that? Because the sensory neuron is way more important. So let's check it out. The sensory neuron is connected to the brain and the spinal cord. We know that if we get any damage to our brain or our spinal cord, we are paralyzed. Did you know that, right?
Yes, sir.
So we say another person, it's because it's over a short distance and the impulses is being carried, so there's no need for a myelin sheet. That's also a valid response as well. Right? So, sometimes I have to think about these things to make you remember it. But in another case, we could say the sensory neuron it needs that protection. More than anything else, it needs protection. So the sensory neuron is where all of them need protection. But that one specifically will need more protection than the rest, to be honest. Especially more than the relay neuron. So relay neuron is over a shorter distance. It doesn't need a myelin sheet. That's also a valid response. Yes. What's a synapse though? And this is very important to note that nerve cells never touch. Did we know that?
It's there, it makes sense that they
Right.
wouldn't touch.
Never touch. Right? So the synapse, what would you say? You say it's the site of transfer. What's a synapse? Or the synapse, some people say it. Call it synapse.
So I think it's where the chemicals are released.
Where the chemicals are what?
Released.
Released. Yeah, it is where the chemicals are released. Right? So what happens is that the chemicals are released here. What are these chemicals? These are the transmission. So let's say we're talking about the heat. Cool, we're talking about heat. We're transferring the heat. Now, it's sending a message to the relay neuron that there is heat here. So what's going to happen now that the messages are going to jump from one side to the other? They're going to jump, right? So they're going to jump from here to here, but they never touch. And the synapse will be the site or the space between two cells where information is exchanged or electrical impulses are exchanged. Right? So we could say it's a site, we could say it's a junction, right? Because you have to say the space or the junction where two nerve cells basically transfer electrical or chemical impulses. Or we could say it's a gap. So gap, junction, space, whatever suits you can work.
Sir, what topic or part of the syllabus is this under?
Yeah, I don't remember, to be honest. Don't remember which part.
Okay, because I don't think I've done nerves at all, to be honest.
Uh, it's there though.
Yes, I know it's there.
Yes, it's there. Since we're the dendrites and neurons are exact. Okay. So yes, we can use those, as long as that makes sense to you, Williams, it makes sense to me, but as long as you can remember those terms, then sure. Then now that heat is transferred to the motor neuron, and the motor neuron would be carrying it to the effector. And what would be the effector here?
Sir, the part of the organism that responds to the stimulus.
So would you say the effector is the same is the same organ as the receptor? Would you say that?
No.
Sure.
Yes.
What's the
Yes,
difference?
but they have different functions.
All right, so let's say, for example, you step on a nail, right? If you step on a nail, you're gonna basically chew off your foot quick, no?
Yes, sir.
Yeah, so I mean, so that effector will be the one that makes you move off that nail.
Yeah. So, sir, the effector would respond to the, where would I say it?
It responds to the message
Yeah. Say you step
or
on
the
a nail.
electrical impulses, yes.
So the effector responds to the stimulus.
Yes, the effector responds to the stimulus.
Is it similar to when something goes in front of your eye really fast and you blink, like you close your eyes? It's similar to
Yes,
that?
it's kind of similar, kind of, but not.
So
Okay,
Because
fair.
why is it not? Because the eyes are involuntary movement. The eye will automatically be blinking.
So if we have our foot on the nail and we take it off, do we really think about taking it off? Like, isn't it kind
No,
of
we don't.
involuntary too?
No, we don't. But no, we don't. But I don't blink because there's danger. I just blink because it need to moisturize whatever the organs are in the body. But if the eye blink, whether there's danger or not, but your foot on a nail just responds to the danger. You get the reasoning?
Yes, sir, I do. Thank you.
All right.
So then, sir, in that case, the receptor and the effector would have to be different because, okay, you're saying that the receptor would be the skin.
No,
Then if we move, then that's the muscle. It's not the skin that makes us move.
it's not the same.
Okay.
Yes. I'm asking all these questions to see how you respond to it.
Sir,
Because
sir,
a lot of times teacher will say something and students just accept it. So in my classes, sometimes I say something deliberately wrong on purpose. And then see how you react to it. Do you sit and take it? If you're not sitting and taking it, it means that you're learning and you're reasoning.
Sir, could you go over neuron one more time? Like, was it?
Huh? Repeat that? Oh, I seem like she's out. All right. So we understand it's not the full, but it's basically the main part of of the nerve system, the diagrams. Let me see if I can take this off the screen. How about simple reflex action? What are reflex action?
Sorry, sir. My data went away. Did you explain um the receptor neuron? I think is what I asked you to explain.
Reset. No, I didn't hear that question.
Oh, can you go back up to the diagram, please? The
This
relay
one?
neuron. Yes, the relay neuron. Could you tell me what it does again, please?
All right, so the relay neuron, what is the relay? What does relay mean?
Sir, I think of like sports running.
Uh, what when in a run, what's a relay race?
When you give someone the baton and you take it back, like yeah, you give someone the
One
baton and
race,
you, yes.
right? So in this case, the relay neurons facilitates communication between a motor cell and a sensory neuron. Sorry. It facilitates communication between a sensory neuron and a motor neuron.
Oh, okay. So just giving information.
Right. So it's passing on the information from the sensory neuron to the motor neuron. So we say that there's fire here, there's heat here, and we say it telling the body, say, hey, there's heat. And then it passed on that information to the relay neuron. The relay neuron passed on to the motor neuron, and the motor neuron passed on to the effector so you can move yourself out of danger.
Okay, so thank you.
No problems. And we say I would say that information travels in one direction only. Our electrical impulses travel in one direction only. We can see it here. One direction only. Right? Because for it to travel all around, you need a relay neuron here, and then it wouldn't make sense because none of these are neurons. They are receptors and they are effectors. So it only travels in one direction only. And then we state that a reflex action. What is that though?
So
When you
sir, when you
automatically do something, but you don't know, so you basically are involuntary
It's an
action.
involuntary action. So my question to you is, does that involuntary action involve the brain?
No, it doesn't.
Yes,
So
I
what?
think it involves a certain part of the brain. So would it be the brainstem, or is it just the spinal cord?
Okay, so some person said no brain, just the spine. One person. So you're saying the stem, but not the actual brain.
Isn't the brainstem a part of the brain, sir? Isn't it like the cerebrum, cerebellum, and then the brainstem?
Technically, it is a part of the brain, but
Yeah.
the relay neuron doesn't involve the brain because of why doesn't it involve the brain?
Because that would take up too much time, and the
Too
reflex
much
action has
time.
to occur very quickly. So I'm
Exactly.
assuming it's just the spinal cord, then, sir.
Just the spinal cord.
All right, thanks.
Yes. Are we clear?
yes, sir.
What's an example of a reflex action?
Knee jerking.
Knee
Knee
jerk.
jerking. Excellent. All right. What's another example of a reflex action?
Sir, I think I said it earlier when I mentioned the eye thing.
The
When you close
eye
your eyes. Yes.
in response to what stimulus?
Something coming quick towards your face or something like that.
Think about it. Think about it, Lee. What makes you close your eyes immediately?
Would it be
Sir,
like
dust.
like
Dust?
if perfume spray, like
Yes,
somebody
dust is one. What? Go ahead.
Like perfume, like if you're spraying your perfume and it accidentally caught in your eye.
Yes. But what is a more common one that I thought you would have said already?
Sir, blinking.
What's one stimulus that's all what you're saying is correct, you know. But what's the most
You're
popular
like when
stimulus?
somebody try slap you
Right,
in your
right.
face or something.
Yes, that's one. But what's a more common stimulus you learn? The whole aspect of it is biology.
Right?
Light,
I just said it.
light. Like
Sarah was
I
saying it.
just
just
said a
imagine
bright
you in a
light.
dark room and they just turn on the lights. So
Like,
all of
sir,
a
when you're
sudden,
waking up in the morning and your mother turn on the lights and you just
yeah.
the light, yeah, touch your eyes, so bright light.
So let's go deeper into that now. Why do you respond to light that way in the dark? Why is it that you have to immediately close your eyes when
Sir, is it because your pupils don't have enough time to close up so to lessen the amount of light that's entering your eye?
oh my God, that's a beautiful answer. If you could get the one now, but I get.
Or just adjusting, like you just adjusting the light.
But I get the one now. Is Dejane? That's your name?
Yes, sir, it's Dijane.
Dejane. My apologies, Dejane. I told you I'm Herbert names. Dejane, that's a lovely answer. So what happens is that when there is, all right, so if you're in the light and you lock it up immediately, all of a sudden the place is pitch black, not true? Can't see.
Yes, sir,
But
your eyes.
as soon as 5 minutes after you start to see little better things, don't you start to see things a little bit more clearly.
Yes, sir. I
And
think
it's
because your pupils dilate to.
exactly. Exactly. So your pupils will open up, right, to take in more light when you're in the dark. But when you're in light, your pupils will constrict in order to restrict the amount of light. Cool. So when the pupils are constricted and you turn off the light, automatically you're not gonna see. And you can't see because there is too little of light going into your eyes. So your pupil will dilate and open up, right? But when the light comes back on your dilated pupils, your eye have to close back immediately because it gives you time, it gives your pupils back the time to readjust to the light that you're in. Spanish, everybody understand.
Understand.
Excellent. So a little bit comfortable with the nerves now.
Yes, sir.
Yes, sir.
All right, so let's jump back to the question. Are you tired?
Hungry.
No.
All right, guys, just soon close off. Because we know they're probably tired. All right. Name one feature that would enable a scientist to distinguish a spherical bacterial cell from an animal cell when viewed under a microscope. So what is one feature that would distinguish a bacterial cell from an animal cell?
The
Sir,
bacterial cell lack a nucleus.
The bacteria has what?
The bacterial cell lack a nucleus.
Lack a nucleus. Beautiful. Yes, what else?
So the bacterial cell would have a flagellum, while the animal cell would not.
Oh, nice answer, sir. Yes, sir. What
sir,
else?
would it be the bacterial cell will have the capsule and the animal cell will not?
Yes, 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 yes. All correct. So we are in the chat and you're all correct also. Nice. One will have a nucleoid and one wouldn't. Nice. All right. And then I just drew it here and explained it. So you should be able to explain and draw, be ready to draw, and be ready to can what's that word again? Label. All the cells, including the bacterial one as well. And my favorite part of the day, genetics. Think you can take on this?
Yes, sir.
Yes, sir.
Are we too hungry?
No, sir.
All right. So figure 3 is incomplete diagram of meiosis in an animal cell. 4 genetically different daughter cells, P, Q, R, S, are produced. First of all, what's the difference between meiosis and mitosis?
Um,
So meiosis,
Sir,
it occurs when it occurs in
All
when you
bodies.
have like male and female gametes, sir. So like
Mhm.
sexual reproduction, and then
Right?
mitosis occurs in asexual reproduction.
We can take that. Anybody else?
so I'm not sure of which one of them, but one of them, I think a meiosis, when they replicate, they have 23, and the mitosis have 46 chromosomes. I
All
think.
right. All
It
right.
Mitosis occurs in all body cells except in the formation of gametes.
Yes. And that is very excellent to know. Meiosis is sexual reproduction, and anything that has to do with sexual reproduction, gametes are eggs and sperm cells, are ovum, are ova and sperm and sperm cell, egg or sperm cell, right? Gametes. Anything that has to do with sex, it's meiosis. Anything that has to do with replication of cells identically would be mitosis, right? Mitosis is identical right down to the T, right? So in meiosis, we have, somebody correctly said that we have 23, we have 23 from each parents, right? So mother and father come together, right? Having sex, then when they have sex, they have 23 from each. So 23 chromosomes from the father, 23 chromosomes from the mother. They combine together to give you 46. Right? So therefore, with meiosis, you're going to have what you call variation. Do you know what variation mean?
yes, sir.
What does variation mean?
Differences.
Differences. That's why all human beings are different, even twins. There are little differences there in their makeup, right? So even though they are the same, they are not genetically the same to the T, right? So there are differences. That's why animals look different. You see, even though we know that dogs are dogs, you can differentiate between two of the same breed of dogs, right? So we see that there are differences in the characteristics of species. Nice. So with mitosis, everything is the same, right? So example of mitosis would be cloning. It's right down to the same. That is why we have mitosis in your red blood cells, because somebody said that red blood cell doesn't have a nucleus. Is that correct?
Yes, sir.
That means that red blood cells would die. That's also die frequently. How often does red blood cell die?
3 to 4 months.
More 3 months. Right? So 3 months are 120 days. 120 days, how much are 3 months? All right, so 3 to 4 months, we'll take that, 120 days. So that means when they die, they have to be replaced. Not true?
Yes, sir.
And we don't see where red blood cells are different. We don't see a different type of red blood cell. All of them are identical. That's why it's mitosis. Skin cells die frequently also. That is why when they die, they need to be recreated through mitosis. Identical. So we see we're all, it's an identical clone. It's identical. Right? So once you understand that, we're basically halfway through genetics. You would be surprised to know, knowing those two, the differences can be a game changer. Then now, we are going down now based on the question. So the question here, we see one chromosome that is a little bit broken, and we see another one that is not broken, arc, and we see one here. Then we say, no, this is incomplete. So we see there is one long one here, one short one here, one U one here, one broken one here. Okay. So it's 4 broken, right? So we see two of the same here. So we said that complete figure 3 by drawing the contents of data Q and R on the diagram. Somebody can draw that for me? You can annotate. You know how to do that?
Sir, I'm not sure if this is correct, but would it be that each of them have a broken line, quote unquote, and another one would have a fixed continuous line?
Okay. So we see one has a broken one here. Jaja. All right. This I got fair enough people head, to be honest. Who never do genetics before? Who not understand nothing about genetics? None at all.
So I did it, but I don't understand.
All right, it's kind of a long topic, so I really want to touch that. Should I touch that now?
No
You feel
problem, sir.
you have the brain power to touch that right now?
Yes, sir.
And that yes, sir, speaks to everybody.
Not so sure.
I'm going to take a vote. Let's take a vote. Because it's complicated, Well, not really complicated, but it will take your brain power.
Sir, it
So,
drains the human body.
so genetics, yes or no, I'm going to create a poll.
Yes.
Oh, boy. The poll will win, guys. Are we seeing the poll? No? Yes? No?
No.
Are you seeing the poll now?
Yes, sir.
All right, so we have 1 for yes, 6 for yes. Ah, man, yes. Oh
Yeah.
my God, you're killing me. Yes, I'm seeing 1 no. 1 no. Oh my, it's
Sir,
14
what was
people.
the question?
Genetics, yes or no? Oh man. 1 no so far. 15 yes. I know. Okay. 17. May I say yes? Yes. What the ones whether I said no? All right, are you?
No, sir. No, sir. I mean, first say yes.
Who said no? Who brave enough to say no? Who said no can say? Who said no? Just put up their hand and say why. Just tell me. Who brave enough? Who said no? I want to remain anonymous. No, I remain anonymous, Sharika, you said no?
Yes, sir.
Sharika, you're brave.
I say no because I maybe have to go away and miss out everything.
Gotcha. Gotcha.
Sir, and it long, it like if you learn it
When
long.
when I started genetics in my bio class, at one point, I was totally lost.
It's simple, man. Simple thing. Simple thing. All right, we're on to it. About our last topic, this though. So I know we have to go to this again. All right, so we're on to it. It's simple though. Simple. Simple stuff. So you're on to genetics. Everybody's favorite subject. Everybody hate genetics. I don't know. Everybody hate genetics. I've never
hate
met
it.
somebody who love genetics.
Sir, I love
I
genetics.
like it.
Only nerd them love genetics like myself.
Don't hate it, but okay.
I'm a nerd. Nothing is wrong with being a nerd, guys. Own up your nerd. Own up your nerdism.
Sir, more to call it nerds, me to call it genius because I actually have knowledge about certain things.
But nerds are
Exactly.
genius.
Exactly.
Nerds are so not like nerds. I'm a nerd.
My classroom is full of nerds.
Really? Which school at?
At Pembroke, there's this one class that every year you have nerds, and that is grade 1146. There is not one person in that class who is not a nerd. Every
Why am
single body in a nerd.
why am I wish me wish the school that I taught were nerds. No.
And and just say that when like that class has about like that class has head boy, deputy head boy, head girl, prefects, head prefect. Like that class is the best class in Pembroke High School. Grade 1146.
Big up grade 1146. Anybody who are watching, them know of grade 1146. Why we can't find genetics here? No idea.
Sir, in the book it's page 145.
145. So far, them put good old genetics. Oh
Yes, sir,
man.
because it's almost the last thing on the syllabus.
All right. So for all the bright people, are you seeing my screen?
No,
No,
sir.
sir.
Oh, you're not. Okay, good. So let's go. So to understand, first we need to understand what are chromosomes. And chromosomes are a what? What are chromosomes? Everybody gone silent.
So
Okay.
Sir, I don't
chromosomes
know.
are like single strands of DNA.
We have single strands of DNA,
They are.
right?
Something like that.
Okay. So there are single forms of strands of DNA. Great. And then we say, do we understand inheritance?
Yes, sir.
Yes, sir.
All right. So are we speaking for everybody?
No, sir.
Yes, sir.
Who will not understand inheritance? All right. I'm going to assume so nobody not understand inheritance. So, single pair of DNA, cool. Then now we say that DNA is deoxyribonucleic acid. Make sure we can know that. And it's wrapped on proteins called histones. Very important for all the nerds out here. Histones. right? DNA molecules contain genetic information in the forms of gene, right? So the genetic information will be in the form of gene. What are some genetic all right, let's not skip. Let's not go there yet. In any cell that is not dividing, chromosome exists as a long, thin strand known as chromatids. Right? So chromosomes exist as long, thin strands known as chromatids when they are not dividing. Hmm? Okay, that will make sense. You know about the phases of mitosis and meiosis?
Yes, sir.
Yes, sir.
Okay, so it's not dividing, it's interphase. Right? It's not really dividing. But them
Isn't it
take out inter.
replicating?
Sir, somebody said no in the chat. I think it's Daniel said no in the chat.
No, for what? What was the question?
The phases, sir. The phases.
Oh, right, because the phases, them they take it out of the syllabus, but they still exist. So in the syllabus, you don't when you're writing, you don't necessarily have to take the phases. You just tell what happened. That's why. So we say chromosomes are passed on from one generation to the next in gametes, right? And each species has a distinct number, blah, 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 blah. All boring stuff. Okay, boring, 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 boring. Let's get down to the not boring stuff. All right, so mitosis, as we said, occurs in all body cells except for the formation of gametes. And we established that. During mitosis, two genetically identical cells are formed, right? And each cell contains the same number of chromosomes as the parent cell, and we call that diploid, right? So every cell contains the same, is identical. So if the same 46 that the mother cell has is the same 46 that the daughter cells will have. Cool? And it's a diploid number. And it's known to be 2n. It's 2n? Yes, 2n. I think, yes. So diploid number are 2n. So anyway, you see 2n, please to note it's not Spanish. It's not Japanese. They're just saying it's a diploid number. It's the same number of chromosome as the parent cell. Are we good so far?
Yes, sir.
Yes, sir.
Yes, sir.
Is anybody not good so far?
Yes, sir.
You're not good, Douglas? Or you're good? Which one?
Sir, I'm not good. I'm not good with genetics.
No, we're not talking about whether you're good with genetics or not. We're talking about if you're good when we explain so far.
Sir, not really.
All right, we could go so again. So mitosis. Remember we said that mitosis is like cloning, right? So if there is thought, let me see how I can put it there now in a good way. Let's say, let's use you, right? Whatever your makeup is, if we are cloning you, you will be the same right down to the T. Right? So if we're gonna make another Douglas, we have to use mitosis because we want you to be the same. Exactly the same. Down to everything. Phenotype, genotype, all the same. We know what is phenotype?
Yes, sir.
The
Yes, sir.
physical appearance of
All the
an
people who
animal.
don't know at the back what phenotype is when you are physically, the physical features of a person. Big eye, big nose, red, brown, black, whatever your color is, that's your phenotype or your physical appearance. And genotype is your genetic makeup. So both your phenotype and genotypes have to be the same. Douglas. So if we want another Douglas, exactly, we're going to use mitosis. Exactly. If we're going to clone you, Douglas, we have to use mitosis because it's going to be exactly the same. How are we understanding so far? You
Yes,
understand,
sir.
Douglas, so far? So Douglas, you are the original Douglas. So you would be the parent cell. Okay? And if we want to, and your
Sir.
clone would be what the daughter cells, right? The cells that
Yes.
are made from you, the copies that are made from you. Think of it as a photocopying machine then, right? You have one original copy of the paper, and then when they are photocopying the paper, it's the exact same paper you see, right? So we said that they have the same number of chromosomes. So Douglas, if you have, for argument's sake, 20 chromosomes, then your clone would have to have how many chromosomes?
So the same 20.
The same 20. And that is called diploid number, or 2n. Is that making sense now?
Yes, sir.
Excellent. Say in a hard, in a hard. So we're going to say now, boom. Why is mitosis important? It's important to ensure that each daughter cell contains the same diploid number of chromosome, and that is 2n number chromosome, because this maintains the species number of chromosome in all members of the species, right? So let's say, for example, we are going to make a community of Douglas, right? So we are going to make 30 Douglas. All of the Douglases must have how many chromosomes?
30.
20. Remember, 20 we use. Right? So remember that the parent Douglas have 20 chromosomes. So that means that if we're gonna make 30 chromosomes, if we're gonna make 30 Douglas, all the 30 Douglas must have the same amount of chromosome, 20. Me lose nobody?
No, sir.
No, sir.
Good. The next important thing is that it ensures that each daughter cell has an identical combination of genes. And what do we mean by combination? We saw back in the question, let's jump back to the question that you were asked. We see that here, if this was mitosis, right? It will mean that all of the chromosomes, right? All of the daughter cells would have to be just like the parent. So everybody would have to look like this. You get where I come from?
Yes, sir.
Yes, sir.
So everybody will have to have the same daughter cells, and the daughter cells will be these chromosomes, right? All the same pairings has to be the same. But we see that there are different pairings here. Cool? Because this is not mitosis, this is meiosis. So if this was mitosis, all of the pairings would be the same. Douglas, are you with me still?
Yes, sir.
And you understand so far?
Yes, sir.
Good. Let's jump back. So we say it ensures that each daughter cell has an identical combination of genes. So it has to be the same. It is the method by which all cells of a multicellular organism are formed. Hence, it is essential for growth and repaired damaged tissue. I remember what I was saying before, right? I was saying that whenever you are exercising, and anybody an athlete here or anybody playing sports? No? Yes?
Yes, sir.
Sir,
What
used to.
what did you
Yes,
play or
sir.
what you used to play?
Netball.
So track
Netball,
and
what?
field.
Track and field. Have you ever experienced injury before?
Yes, sir.
Yes,
Yes.
sir.
And when you're injured, it means that your muscle is teared, right? Or
Yes, sir.
strained.
Our
In
tendons.
order, yes. So in order for that to repair, right, it need to recreate back those exact cells to repair your muscle. So therefore, mitosis is needed for growth and repair. Because remember, you know, when you're a 3-year old, do you have the same muscle as you are now?
No, sir.
No.
Because you get stronger as you grow, don't? So that means your muscle need to grow. What makes it grow is mitosis because of the same cells is going to be replicated down to its identical. So if at 20, they're going to replicate the same. No difference, no changes, the same replication, just more cell. More of the same cell when you grow. That's what growing is. You get more of the same cell. Did you know that? More of the same
No,
cell make
sir.
you grow. Never
Yes.
know that.
Yes, sir.
So more of the same cell make you grow. So mitosis is important for growth and repair. And then of course we know asexual because it form an identical to the parent. Are we good so far with the importance?
Yes,
Yes,
sir.
sir.
Then now we look at the stages, right? We said that the first stage, we see that this is a cell. This is a nuclear membrane. The centrioles are here. And we have the chromatid threads that are little pieces of blue here. And this is when the cell is not dividing and the chromosomes are not visible, right? So they are chromosomes. They are not visible as they exist as long, thin strands called chromatids. So when the cell is not divided, the chromosomes are not visible. They are called chromatids, right? And this is normally in interphase, but then they don't use the phases anymore. So we just say that. Then the next stage, we say that the chromosomes are shortened and thickened and duplicate themselves and become more visible. So we see here that the centromere is like a thumbtack and it joins blue, long and long blue together. It joins short blue together. It joins long red together and it joins short red together. And they are joined by the centromere. And we see where the centrioles form a spindle fiber in between them. So we see where the centrioles move to both poles. Right? Why is it moving to both poles? Because what is going to happen now is that the nuclear membrane is going to disintegrate. We see that there's no more nuclear membrane here. It's gone. And then what happened now is that the spindle is going to form between the centrioles. So we're going to have spindle here. Let me get the annotation. We're going to have a spindle joining here, a spindle joining here, and a spindle joining here, and a spindle joining here. Right? And then what's going to happen now? It's going to pull apart. Right? Why is it going to pull apart? Because it's going to form two daughter cells. Remember that CM2N? It will form another cell. So what happened is it's replicating here. So it replicate, and then what happens is that the spindle fiber now pulls it apart. And then we see when it pulls it apart, it's going to the opposite end of the pole. See, it's going to pull here, here, and here. Let me get a different thing here, here. Cool? And then when it pulls it apart now, we're going to see where there's going to be a constriction in the middle here. Because what's happening now is that the cells the cell is now going to form a new cell. So we see where the chromatids reach the opposite pole and are now called chromosomes. So when the when the chromatids are pulled to the opposite pole, they are referred to as chromosomes. And then we see now where there it forms two identical daughter cells. We see here, we see one long blue, one sharp blue, one long red, one short red, and we see the both of it here. And that is how it is formed. So we have how much steps to remember? 1 step, 2 step, 3 step, 4 step, 5 step. Basically 5 step. Have I lost anyone?
No, sir.
Right? So you'll have to, oh, somebody asked what are centrioles? Centrioles in layman terms, centrioles are like thumbtacks. Centrioles are the things that are there. The centriole forms it forms the spindle fiber that is important to pull apart the chromatids in the third phase. Right? So that is what the centromeres are, Williams. So this is something we definitely have to read after. So you can take a picture if you don't have the book. Right? If you have the book, you can read it. Is there anything you don't understand so far from this diagram here? All good.
Sorry, yes, sir, it's 5 steps, 5 phase. But it's 6 phase. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6.
Yeah, 6 phase.
Okay.
The 6 phase, right? So the other one is cytokinesis, basically. But they don't require you to say that anymore. But if you can learn, if you want to learn it, nobody's stopping you. But they take it without the phases nowadays. So that's what I'm saying, Harriet. It's kind of a mix-up. So you just need these are the steps they need to see, Harriet. If you want to use the phases, you can, but it's not required in the syllabus. So prophase, metaphase, anaphase, telophase, all of them phases are not required by the syllabus for you to know. You just need to know what happens. Right? The phases are now taught in Cape level, at Cape level. That's where the phases are now moved to. So you don't need to know them, but you can. Teachers still teach them, but the syllabus doesn't require you to say them. You don't have to say this is anaphase or this is telophase. You just need these steps are only what is required by CXC these days. Cool? Then now, meiosis is the same. Phases, but meiosis is doubled. Why meiosis is doubled? Because meiosis, let's see if we can skip to meiosis.
Sir, is it double because you need crossing over to occur for variation, sir?
It's double because instead of 2 daughter cells,
It
meiosis is
forms
4.
4.
Yes. So instead of 2 daughters, we see that at the end of the day, we see 2 cells here. True? And they're identical. 2N. In meiosis, it's what we call haploid, not diploid. Is Douglas still here? Douglas report.
I'm
Right,
still
Douglas.
here.
Oh, you're still here? Okay, good. So, unlike mitosis, meiosis now has to do with sexual. So in meiosis, we see that there's going to be 4. But before we move to meiosis, let's finish up mitosis. So mitosis example would be asexual reproduction and cloning. Do you know what's the first animal to be cloned? Have you ever been taught
The
that?
sheep.
One sheep,
And
sir.
what's a sheep name?
Don't remember, sir.
Sally,
Dolly.
Dolly. Yes.
Dolly. Dolly. The sheep name is Dolly. So cloning this first sheep, that's Dolly. We also have vegetative organs, right? Like rhizomes, stem tubers, corms, and bulbs, and all of these. You know what are rhizomes?
Yes, sir.
What is rhizome? Jaja man and gone. Rhizomes are like ginger, things
So
that growing on the
rhizomes are
earth.
like roots that have a lot of nutrients in them, sir.
Yeah, like ginger. Ginger is the most
And
popular rhizome there
not
is.
currant, sir. Carrot.
Turmeric.
Oh, sir, would carrot be a rhizome?
Oh, carrot is a what is carrot again? No, I don't know. No,
All right, sir.
no, no. I think carrot is just a what is carrot? Somebody can check that. I think carrot is just a root, I don't remember. I'm not going to pretend that I remember. So somebody can check that out. I'm going to tell you the safest answer though. I think carrot is a top root, if memory serves me correctly. You can check that out. I think carrot is a top root. But for rhizome, the safest answer and the most relatable answer is ginger. Right? Or ginger growing in the earth. Right?
Yeah, sir. I did a lab with like a drying lab for carrots
Yes.
in bio, and
Yes.
it said the top root was a part of the carrot, like the bottom part of the carrot. So that kind of confused me, but all right, so top root.
Okay. Well, I don't know. A lot of people have different versions, but my version, the version that I study is that it's a top root. Stem tubers would be like potato. Carrot could be a stem tuber, my youth. All right.
Yes, sir.
Yeah, man. Carrot would more be a stem tumor.
Oh, a stem tuber, sir.
Yes.
Okay, because it's like a tuber, but it has a big stem and shoot growing out of it.
Yes, sir.
Okay, thanks, sir.
Yeah. So now that I'm seeing it, so rhizome with ginger, stem tubers will be more carrot and potato. Right? So all of these are example of mitosis because we see where they are similar. You ever seen a ginger look different from the other? Not really. They might look different in sizes, but they are the same. Right? And new plants can grow from another plant, like adventitious roots. Where you see runner grass. You know them grass the way jump out I and them just long and them just connected. All of them they are examples of mitosis. Right? So we're gonna skip the tissue culture and now run up in a meiosis. For the time, let me know, you know. Hmm. For the time, let me know. But still strong?
Yes, sir.
All right. So for meiosis, we see where meiosis occurs in reproductive organs during the products of gametes, during genetically non-identical cells are formed. Right? Each cell contains half the number of chromosomes. Right? So then we go back to the mother have 23, the father have 23. Right? And they are known as haploid cells or N number. So what do we say? Mitosis. What type of cell mitosis? What type of number mitosis have again?
Diploid, sir. 2N.
Diploid or
2N.
right. And then meiosis has what kind of number? What kind of number?
Applied are in.
4.
N number.
Right.
Haploid number
Haploid
or
or
N.
N number. Exactly. So why is meiosis important? Meiosis is important for one very, very, very important reason, and that's variation. Variation. Why is variation important?
So variation is important to help a species adapt to changes in its environment, sir.
Yes, sir.
It's helpful to carry positive genetic traits that help the species survive.
Okay. Anybody else? Is something slightly different or same? All right. So in the interest of time, we'll say that variation is important to prevent starvation. And why our genetic variation prevents starvation?
Don't know, you know, say.
Because if you have everybody of the same type, they might eat the same food, then there are going to be no food.
Never know that they one there, sir.
Think about it. If all the organisms eat, let's say there is genetic clone, let's say there are 600 cow in one avenue, then what kind of grass are going to there for them? All them might eat the same grass. They never going to end up dead. They might end up fight, right? There's going to be more competition for the same food, right? Then eventually they are going to die out.
So
They
this is
cannot,
probably oh,
you go ahead.
so this is probably wrong, but is uh is variation why we have like omnivores and carnivores and all that?
Yeah, basically, because yes, 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 indeed. And also, we can look at, let's say, for example, there are 700 cow genetically make up the same. And one disease come and kill out all cows that kill the same cow. And all I call them might then have the same disease. Right? Like COVID came and hit us. Some people die, but some people survive. Why some people survive? Because their genetic makeup is different and they could fight the virus. But if you have everybody have the same genetic makeup and it kills one person and everybody of that genetic makeup would die. Make sense?
Yes, sir.
Yes, sir.
Excellent. So variation is very important and come on to that. So we can see here where it's the same. It's the same basically, where we have the two chromosomes of maternal origin, right? And we see the centrioles at the end just the same. So the nuclear membrane is right here. And we see there are two chromosomes of paternal origin. So the red one is from the mother and the blue one is from the father. And we see where the chromosomes are shortened and thickened and become visible. Then we see where they pair together. The long one is paired and the short one is paired. We look at mitosis. We see where the long one is paired with the two long blue are paired together and the two short blue are paired together, just like the red. Right? So we see the differences already. So when we do the thing, we have to make the differences known. Right? And we see where the central are separated and start to form the spindle um fiber. When the two long one pairs, they are not pairing together, they are pairing separately. So we see where blue and blue pair together with mitosis because everything has to be identical. But here we see where they are not paired. They are just paired by the length and not by the color. You see that?
Yes, sir.
Yes, sir.
I will call that bivalent. So something that's added in a nerd category, bivalent. So when you're bridging them attack and them a run up in your face, ask them what bivalent is. You see me? That's how we nerds do it. So we'll go down and we see where there's a crossover point. So we see where the centriole is in the middle here. It joins them together. It joins blue and blue, back, red and red. Just like how it is here. So blue and blue long is joined, red long is joined. But the only difference is that they touch. They are not separate. They touch because why do they touch again?
Is it because of crossing over?
And what is the crossing? What do we mean by crossing over?
Hold on.
Crossing over mean that they share genetics. So, so you see where after they cross over, you see where blue has a little bit of red and red has a little bit of blue, vice versa. You see that?
Yes, sir.
Yes, sir.
Because they are sharing information. When they share information, they are sharing information to not be the same. See that?
Yes, sir.
So
Sir,
we
so
say,
what, excuse me,
yes,
sir.
yes, go ahead.
So what you're saying is that they're exchanging genetic materials.
Yes. Are we all clear?
Yes, sir.
Yes,
Yes,
sir.
sir.
Why that group of brides still, you know? We check some of the face out already. Given that we didn't start with genetics. Because normally when I'm doing a workshop, I start with genetics. Like I would do a workshop just on genetics. On a strong man. On a strong. All right. So then we see where they are separated. Just like how they they did in the mitosis. And we see where they separate. But they separate in homologous pairs. What does homologous mean?
The same.
Same. Excellent. So they separate homologously. So we see here, instead of two daughter cells, we see how much daughter cells?
4.
Four. So we see four in each. Good?
Yes, sir.
And we call this phase prophase 1. We call this mitosis 1. Right? So this will be, so the phases will be prophase 1, metaphase 2, metaphase 1, anaphase 1, telophase 1. So this is phase 1 basically. And then what's going to happen now is the same thing is going to happen over here to get the 4 daughter cells. So we see here that when they separate into 2 pairs, what's going to happen is that the 2 cells is going to replicate. And the 2 of them is going to replicate with them exchange DNA. They are going to join up. Then the spindle fiber is going to form as usual. And then now the spindle fiber is going to pull them apart. So we are going to see that 4 cells are formed with two pairs. See that? And these 2 pairs have a little bit of, you see that blue here as a red, and this red here as a blue, so this genetic information is shared.
Sir,
So yes.
okay, you probably went over this already, but I didn't quite catch it.
No problem.
Why does meiosis have 2 phases?
It has 2 phases because it's going to form haploid number, and a haploid number is 4 cells. So it's going to form 4 daughter cells. So we see where the first phase have 2 daughter cells that's going to form here. We see that?
Yes, sir.
And normally this is where mitosis would end with a diploid number, but for meiosis, we want a haploid number. So there's going to be, we need 4. So that's why now for phase 2, these 2 cells that were created here, right, is going to further be replicated again. So we see these same 2 cells here are being replicated. So the 2 here, 2 each here, is going to be replicated. Right? And we're going to see where the same thing is going to happen. You're going to pull it apart, and it's going to form 4 daughter cells, which is a haploid number.
Okay, sir. Thank you.
Excuse me, sir, I have a question.
Go ahead, sir.
So directly after the first cytokinesis, sir. So right after telophase 1, is there a next interphase between the cytokinesis and phase 2? Okay.
No, one interface.
Okay, sir.
This is all clear?
Yes, sir.
It is.
No, no, say it now go clear in terms of remembering it, no, but when you read it over, you understand what you read.
Yes, sir.
All right, great. Is there anybody that was still lost in all of this?
No, sir.
There are 20 people here. Some of them not talk. Are the people that's not speaking understanding?
Yes, sir.
Okay, I hope so. Because if you're there, you just look, are you there and gone in your friend house? Are you there and a cook? Might as well you not bother. That's what you're recording on YouTube. Alright? So, now we're gonna go into the fun stuff. So now that you understand all of that, we are now going to go into meiosis and mitosis. And we're going to go into inheritance. Now, inheritance, like chromosomes, genes, I think we could do this question now. Could we do that question now? Could
Yes, sir. I
we?
could.
All
Yes,
right, so
sir.
I think we can flip back to that question and now, you know, one thing.
Yes,
Yes,
sir.
please.
So we can try.
Yes, sir.
Yeah, man, I think we can flip back, man. We could jump back on the big question now. What would Q be looking like? And what would R be looking
Sir, I
like?
think you would have one long strand that is broken, made up of broken lines, and the small line would be unbroken.
So you say Q, and if you want to annotate, somebody can if you want to. I give you free rein to do
All
so if
right,
you.
so I annotate.
So while Harriet annotate, anybody else want to tell me what you think gonna happen in Q?
Sir, would it be, well, I have the
Mm-hmm.
one short
Mm-hmm.
one short broken line and one long unbroken line.
Yes, that is good.
Okay.
What would R be looking like?
For R, I have one long unbroken line. No, one long broken line and then one short unbroken line.
Yeah, man. Dejanay, you're learning.
So, sir, hold on.
Yeah.
Sir. So, is the one that I have in Q incorrect?
So you have one long broken line and
Yes,
what?
sir. And one short unbroken line. Just the opposite of what
No, 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 no. It's fine. As long as you're,
okay. Okay.
it's fine. It's not necessarily one for one. It's anyone and it will be fine. Because because they are looking for the concept. There's not one specific cell that's going to have something different, really. Based on what I'm seeing. Because one have two broken, long, short, da 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 da. Yeah, right. Yeah. Excellent. Is there anybody that's lost to this? You see where this come about? No, yes, maybe so. The people may not talk, are you lost?
Sir, I'm good.
That's Douglas.
Sir.
Okay. Anybody else good? Everybody else good?
Yes, sir.
Let me know if you're not good enough. Tell me no want to chat me, If you chat me, let me explain that. I'm not eating a dinner, I'm not eat no dinner yet. So if me the past when I something name fasting, at least I know me fast and learn. So if you wanna learn, let me know. Me go by it 100 times. I'm not discriminating. I just want some ones to make the society better and the Caribbean better. And that sound good.
Yes, sir.
Yes, sir.
Mm-hmm. So we're understanding. Great.
Yes, sir.
All right, so let's look. So this is something what you look like. Can you about your annotation, right? Oh, I can do it. Cool. So that is what we'll be looking for. Make sense?
Yes, sir.
Yes, sir.
All right. So state whether daughter cell produced by meiosis are haploid or diploid.
Sir,
So,
it's a haploid.
Haploid.
all right, what you done? One name I learn. Killer.
What?
Yeah, name one part of the body in which meiosis occur.
Ovaries
Reproductive
of
organs.
females.
So, the womb or the uterus?
In the
Are the
Uterus.
old
ovaries.
Are
In
the
the
what?
reproductive system.
The ovaries.
Alright, ovaries. Cool. So we have the gonads for the nerds. Gonads.
Sir, so
Yeah.
if me have reproductive organs, is it wrong?
No.
Okay.
But we just want bossy. You see me? You
Okay,
want
sir.
bossy. You know, want just bossy. Everybody say that reproductive organ, you're writing gonad. And then some people probably ask you, what are gonads? And you say, bro, it's testes and ovaries. Come on, why are you writing two words and you can write one? That's the thought of swag you should be having.
So, sir, you're saying that's the swag of a nerd.
Yeah. Swagger for ultimate nerd. And then now name the structure within the nucleus which contains genetic material.
Nucleus.
We say, what go again?
Chromosomes.
Chromosomes. Okay.
Chromatins.
Chromatids. Okay.
Chromatin thread. Sorry, is it chromatin thread?
Yeah, it could be chromosomes. Chromosomes is fine. Chromatids is fine. Oh, yeah, you're talking about when it's ending. So when it's ending, it will be chromosome. So in its when it's not replicating, chromosomes is fine. Okay. Yeah, me feel like say we can call it right there. So I wanna say.
Yes, sir. Yes, sir.
Feel like gonna get the
Yes, sir.
me feel like we touch the genetic part, we have to do that fresh. Is there anybody here that's not a part of the group? Is there anybody here that's not a part of the group? The WhatsApp group. You're not a part of the WhatsApp group? If you're not a part of the WhatsApp group, please to join and
Sir,
you can yes.
I think there's a person in the chat by the name of Jody Ann Robinson. She said she's not in the WhatsApp group.
Okay. So what I'm going to do, I'm going to separate the free WhatsApp group shortly. And I'm going to make one for the serious people. And because when you do free session, not everybody is necessarily serious. So what I'll do is create one that's for persons that are serious over time, and then we can do some work here. So let me just get the link up and going. Why is it that? Okay, so anybody that's not a part of the WhatsApp group, you can join there. And also anybody who need extra classes, we have classes running during the weekend and also we after school like 5 o'clock, 6 o'clock, them time there. We run through topics and these things. And the class is just $1,000, guys, and you get like 2 hours, 2 solid hours.
Excuse me, sir.
Yes. Go ahead.
Sir, I was asking if anybody knows any classes that's going on for chemistry and physics, because I do those subjects and I was asking if anybody could like recommend or tell me about any classes that is happening for those two specific subjects, chemistry and physics.
All right, so I know chemistry classes, but then kind of dear stills, isn't it?
Shakira or Sharika, sorry. Which school do you go to again?
I go to Pembroke High School.
And where is that again?
On Duane Park, Kenil Drive, on the boulevard.
Okay, so there's at my school, St. Andre High School for Girls, there is this teacher called Mr. Burrell. He does chemistry classes and it's every Monday, and the cost is $2,000.
And there's also a teacher at Arden. His name is Mr. Wadsworth. He does it for $2,000 on a Saturday.
On a Saturday, at what time?
Okay, so chemistry is from 9 to 10, physics is from like 11 to 1, and then biology is from 4:30.
And then chemistry at my school is from 3 to 5.
Are those physical classes or you have to
Yeah,
go?
physical classes.
Physical classes, sir.
Okay. But
Okay.
there goes, there you
By
go.
by chance, any one of you guys can text me on my phone number in the group.
Which one is it?
Rika.
Hold on.
Okay, guys. So,
And thank you
yeah,
so very much. It helps.
no, yeah, man. So as I'm saying, the classes them kind of the price them kind of rule still. So if you can, that's a $2,000 for the hour or $2,000 for the week. How that go?
$4,000 for a session.
And a
Yes, sir.
session is an hour.
A
Not
session is
necessarily.
2 hours.
Or a session
So,
is 2 hours. Okay, I not bad.
sir,
Yes.
also, although it's in May, there are workshops in May for chemistry and physics. It's from 3 p.m. to 5 p.m. and they'll go through theory reviews, exam preparation techniques, and also past paper questions. So for chemistry, it's on Tuesday, May 13, and for physics, it's on Tuesday, May 20.
And which school is which company is doing that and which teacher is doing that?
There are a number of universities that is included, and it's a virtual STEM CSEC workshop.
Oh, that one's on the Scientific Research Council. That
Yes,
one?
sir. Along with the Ministry of Education,
Oh yeah,
UTech,
yeah, yeah.
and the University of the Commonwealth of the Caribbean.
Yeah, man, I know that one, but that one is normally crowded still. Like,
Oh, okay,
yeah,
sir.
kind of, yeah. Anyone else, the ministry that one, it kind of crowded. It's like this one now, you can like, you can ask some question and anything like that. That one like 500 kids. So, yeah, sometimes some kids get lost stuff. For those ones, if you're gonna join them, you would want to, you would want to be vocal. So I encourage you to be vocal with them. All right, so make sure say you not care about nothing, you be vocal and state where you not get. All right.
Yes, sir.
All right, so everybody make sure say we keep the link, keep the touch. Remember my classes are like $1,000 for 2 hours. I'm basically giving you a life to freedom. So I'm giving back.
Okay, sir. On
Then
on what days?
on Saturdays, well Saturdays, sometimes Sundays and during the week, depending on the groups.
Okay,
Yeah,
sir.
and
Do
that's
you cater for Cape students?
Yes, I do.
Sir, I'm planning on doing that this year and the next year.
Yeah, Cape student are getting a love with biology, me. I try to give them some love. Like everybody
Okay,
forget Cape
sir.
student. Yeah, for real. Like last year, me have a lot of Cape students, but them they don't really get love. Like all CSEC get love. They don't really get that much love. But anyway, guys, thanks for coming out. Make sure you're in the group. And make sure we keep the link and make sure you're there for the next workshop. I hope you enjoyed this workshop and it wasn't boring. I want to
yes.
enjoy yourself.
Okay, sir. Thank you.
I want to
Yes,
learn
sir.
something.
Bye.
Thank you.
All right, guys. Now those on
All right. Bye,
YouTube,
sir.